Welcome back to the BioCurious podcast. I'm Iode, and today we're diving deep into the fascinating world of proteins. Sharon, I'm so excited to pick your brain on this. Proteins are everywhere, right?
Absolutely, Ayode. It's a pleasure to be here. Proteins are, without exaggeration, the workhorses of the cell. They constitute about half the dry weight of most cells, which is pretty significant. They're involved in virtually every biological process.
Half the dry weight. Wow, that really puts it into perspective. So we know they're abundant, but what exactly do they do? I mean, beyond just being proteiny.
Right. Their functions are incredibly diverse. Think of them as the Swiss Army knives of biology. They act as regulators, like enzymes that speed up reactions, or hormones like insulin that control blood sugar. They're part of our defense system, like antibodies fighting off infections. They're catalysts. Pretty much every biochemical reaction in our body is facilitated by an enzyme, which is a protein.
Oh, so enzymes are proteins. That makes so much sense. I always picture these little molecular machines. What else?
Exactly. They're like tiny machines. Beyond that, they're transporters, moving molecules across cell membranes or even through the bloodstream, like hemoglobin carrying oxygen. They provide structural support, think collagen and connective tissues or keratin in your hair and nails. They enable movement, like actin and myosin in muscle contraction. And they're crucial for signaling, acting as receptors to detect messages from outside the cell. The list just goes on.
That's an incredible range of jobs. It's clear why they're so vital. But for them to do all these different things, they must have some pretty intricate structures, right? How does that even work?
You're hitting on the core concept, Ayode. The structure of a protein is intrinsically linked to its function. It all starts with the primary structure, which is simply the linear sequence of amino acids linked together by peptide bonds. Imagine a long chain of beads, where each bead is a different amino acid. There are 20 common amino acids, and their specific order in that chain is absolutely critical.
A chain of beads. That's a great analogy. So the sequence is the first level. What happens after you have this long chain?
Well, that linear chain doesn't stay linear. It starts to fold into more complex shapes, and we describe this folding in four levels of structure. Primary, as we just mentioned, is the sequence. Then we have secondary structure.
Secondary structure. What kind of shapes are we talking about here?
Here, the polypeptide chain starts to form localized repeating structures, primarily stabilized by hydrogen bonds between the backbone atoms, not the side chains. The two most common forms are the alpha helix and the beta pleated sheet.
Alpha helix and beta pleated sheet. I've heard those terms before. Can you paint a picture for me?
Certainly, an alpha helix is like a coiled spring or a spiral staircase. It's a right-handed coil with about 3.6 amino acids per turn. The hydrogen bonds form between the carbonyl oxygen of one amino acid and the amide hydrogen four residues away, holding that spiral shape together really tightly. The R groups, the unique parts of each amino acid, point outwards from the helix.
So it's like a neatly wound coil, and all the little sticky outy bits are on the outside. And the beta pleated sheet?
The beta pleated sheet is more like a folded fan or a corrugated sheet. It consists of several beta strands lying side by side. These strands are held together by hydrogen bonds between their backbone atoms, forming a very stable, often flat zigzagging structure. These strands can run in the same direction, which we call parallel, or in opposite directions, known as antiparallel. Antiparallel sheets tend to be a bit more stable with straighter hydrogen bonds.
Okay, so we have the initial sequence, then these localized coils and sheets. That sounds like a pretty good start to a complex shape. What's next?
Next is tertiary structure. This is where the whole polypeptide chain folds into its unique overall three-dimensional globular shape. This level is stabilized by interactions between the R groups or side chains of the amino acids, which can be quite distant from each other in the primary sequence.
Ah, the R groups get involved now. What kind of interactions are we talking about with those?
Precisely. You have hydrogen bonds between polar side chains and salt bridges, which are ionic interactions between oppositely charged side chains. Crucially, you also have the hydrophobic effect, where nonpolar R groups tend to cluster together in the interior of the protein to avoid the watery environment. And then there are disulfide bonds, which are strong covalent bonds between two cysteine residues. These are like molecular staples, providing significant stability.
Disulfide bonds. That's interesting. So the sulfur atoms in cysteine link up. It sounds like a lot of different forces working together to get that perfect 3D form. And then finally, quaternary structure?
Bingo! Quaternary structure is present when a functional protein is made up of multiple polypeptide chains or subunits that come together. Think of hemoglobin, the oxygen-carrying protein in your blood. It's an oligomeric protein, meaning it has multiple subunits, specifically 4 polypeptide chains, 2 alpha and 2 beta chains. These subunits interact with each other to form the final functional complex.
Oh, so it's like a Lego structure, where each polypeptide chain is a separate Lego brick, and they all fit together to make the final functional model. That's really cool. So when scientists want to figure out what a protein does, they first need to know its amino acid sequence, right?
Exactly. Determining the amino acid sequence, the primary structure, is foundational. It's a multi-step process. First, you need to know the composition, what amino acids are present and in what quantities. You do this by completely breaking down the protein into its individual amino acids through acid hydrolysis, then separating and quantifying them.
Makes sense. Get all the ingredients laid out. But knowing the ingredients doesn't tell you the order. So how do you start figuring out the sequence?
That's where in-group analysis comes in. You need to identify the amino acid at the beginning, the N-terminus, and sometimes the C-terminus. Methods like Sanger's method or, more commonly, Edman degradation are used. Edman degradation is particularly clever because it sequentially removes one amino acid at a time from the N-terminus, identifies it, and then you can repeat the process on the shortened chain. It's like peeling an onion, layer by layer.
Ooh, peeling an onion. I love that. So you can figure out the first, say, 50 or 60 amino acids. But what if a protein is hundreds or thousands of amino acids long? You can't just keep peeling.
That's precisely the challenge. For larger proteins, direct sequencing of the entire chain is impractical. So what we do is we break the protein into smaller, overlapping fragments using very specific molecular scissors called endopeptidases. These enzymes cleave peptide bonds at very specific sites. For example, trypsin always cuts after a lysine or arginine residue.
Okay, so you selectively chop it up, but how do you know how these chopped up pieces fit back together?
That's the trick, Oede. You use 2 different cleavage methods. So imagine you cut a long sentence into words using a red pair of scissors. You sequence all those red fragments. Then you take a fresh copy of the same sentence and cut it with a blue pair of scissors that cuts at different places. Now you have a different set of blue fragments. By finding identical overlapping sequences in your red and blue fragments, you can piece the entire original sentence back together.
That is genius. So the primary structure is foundational. It determines everything. But what happens if something goes wrong with the folding? I've heard of proteins denaturing.
Denaturation is a critical concept. It's when a protein loses its native, functional three-dimensional shape. This loss of shape usually means a loss of biological function. The primary structure, the amino acid sequence, typically remains intact, but the secondary, tertiary, and quaternary structures are disrupted.
What causes that to happen? Like, what pushes a protein to fundamentally change its shape?
Many things can cause it. Heat is a common one. Think of cooking an egg. The clear starts to turn white as the proteins denature. Temperatures above 60 to 70 degrees Celsius can disrupt those weak interactions like hydrogen bonds and hydrophobic interactions. Changes in pH are another big one, as they alter the charges on amino acid side chains, disrupting ionic bonds and hydrogen bonds. Chemical agents like strong acids, bases, urea, or detergents can also wreak havoc. Even mechanical stress, like vigorously whipping egg whites, can denature proteins.
So, if a protein denatures, is it just done for? Like, can it ever go back to its original shape and function?
That's a fantastic question, and it was the subject of a Nobel-winning experiment by Christian Anfinsen in the 1950s. He worked with an enzyme called ribonuclease A, which needs a very specific 3D shape and several internal disulfide bonds to function.
And what happened? Did it stay denatured?
Here's the remarkable part. When he carefully removed the denaturing agents, the ribonuclease A spontaneously refolded back into its original active three-dimensional structure, and its enzymatic activity was fully restored.
Whoa, so it can refold itself. That's mind-blowing. It's like an instruction manual is built right into the sequence.
Exactly. That was Anfinsen's groundbreaking conclusion. All the information required for a protein to fold into its correct native 3D structure is encoded solely within its primary amino acid sequence. It's a foundational principle in biochemistry. The folded state is the thermodynamically most stable conformation.
That's so profound. It really underscores why understanding the sequence is so vital. And I suppose if anything is off in that sequence, it can have massive consequences for the function.
Precisely. The structure dictates the function. Take myoglobin, for example, a globular protein that stores oxygen in your muscles. Its compact, soluble structure allows it to bind oxygen efficiently. Contrast that with collagen, a fibrous protein that forms the triple helix, providing incredible structural support in your skin, bones, and tendons.
Totally different jobs, totally different structures.
Right. And if there's even a single amino acid change, it can be devastating. Think of sickle cell anemia, where a single amino acid substitution in hemoglobin causes red blood cells to deform, leading to serious health issues. Even diseases like Alzheimer's or Parkinson's are linked to misfolded proteins aggregating. Understanding protein structure-function relationships is absolutely critical for medicine, drug design, and understanding life itself.
What an incredible journey from a simple string of amino acids to these complex, functional molecular machines. Sharon, thank you so much for breaking down protein structure and function for us today. It's been incredibly insightful.
My pleasure, Oeda. It's a truly fascinating field.
And thank you to all our listeners for joining us on BioCurious. We'll be back next time with another deep dive into the wonders of biology.